It's my pleasure today to introduce Shane Palmer. He's giving his exit seminar. He's working for Dr. Van Yersel, but Mark couldn't make it today, and so I'm just filling in to introduce Shane. I'm in his committee as well, so I thought I could take this opportunity, and I'm happy to introduce him. Shane has had a very vibrant academic background. He has a double bachelor's. He has a bachelor's of art degree, and I didn't know it was in fine art until today, but from University of South Florida. And then he has a second bachelor's degree in science in biology, and that again is from Florida. And after that, Shane also did an internship in NASA, trying to understand crop growth and physiology under controlled environmental condition, more analogous to like the space flight conditions. And after that, he came to UGA, and he has been doing his master's under Dr. Van Yersel. And today he will present the work that he has done with Mark. And with that, Shane, you can start.
Okay, thank you. Let me share. Here we go. So good afternoon, everyone, and thank you for attending my exit seminar. I'm happy to present my research today that I've been doing for the last few years, working with Dr. Mark Van Yersel. Today, I'm going to talk to you about a series of projects that I've worked on involving photoperiodic effects on growth, photosynthesis, and biomass allocation in leafy vegetables in controlled environments. So just as an overview of what I'm going to do today, I'm going to provide a general introduction to the problems that we hope to solve.
Shane, sorry to interrupt you. We can't see you. Is your camera on?
This moment, should I have my video up? Is that preferable?
Yes, I think that's preferable if we can see who's speaking.
Okay. All right. So I'm going to give an introduction to just the overview of some of the current problems that we're hoping to solve. And then I'm going to go into my 3 studies that I conducted. And I'll have a moment at the end of each of those 3 studies for some questions. And then I'll wrap up with conclusions and give my acknowledgements and any final questions at the end. So as everyone attending, I'm sure, knows, photosynthesis is a fascinating process where plants are able to use light energy to fix carbon from the atmosphere and accumulate biomass. And whole plant photosynthesis at the whole plant level, not just at the leaf level, is directly related to overall crop productivity. And one of the things that we're focused on in Mark Van Yersel's lab is looking at how can we improve the efficiency that plants are able to use those photons of light to produce marketable biomass. And when we're successful at this, we can help reduce input costs and increase yields for growers. This is especially important in controlled environments, such as greenhouses and indoor plant farms that use supplemental and or sole source lighting for their plants. In these contexts, electricity for horticultural lighting can be up to 30% of growers' recurring costs. So we're fortunate right now that there are a lot of new technologies that we have access to, such as LED lighting fixtures that can be precisely dimmed. We have programmable control systems and smart systems. And these are all ways that we can have a better controlled and more precise timing and intensity of light. In addition, with the new technologies, especially with LEDs, we have a lot more options as far as spectral variability goes as well. So as I mentioned, some of the applications of this research are to reduce the cost of lighting for growers, potentially to extend growing seasons as well, especially in northern climates where seasonal changes in photoperiods happen, potentially shorten crop cycles if we can get the plants to grow faster, and also alter plant growth morphology and maybe phytochemical content as well. So just for some terminology so everyone's on the same page here, when I talk about photoperiod, that is the duration of time that a plant receives photosynthetic light during the day. And a related term is photosynthetic photon flux density, which I'll refer to as PPFD, which is basically, it's a measurement of light intensity, and it's an instantaneous measurement. And it's measured in micromoles of photons per square meter per second. Another related term is daily light integral. So PPFD is an instantaneous measure. Now, daily light integral, or DLI, is the integrated PPFD over that 24-hour period. So that's a cumulative measurement of the entire amount of light that a plant receives in a given day. And that's measured in moles of photons per square meter per day. And this is a visual representation of how PPFD is calculated. The little colored sprinkles that are falling down onto the square mat there are photons. And this is sort of like a flow rate. It's basically a flow rate. So we're counting photons as they're hitting a set unit of area over a set period of time. In this case, it's per second. And to give you an example of how DLI fits into this, this graphic is an example of the light intensity over time on a day. And if you remember from your calculus class, if you take the area under the curve, you've just integrated. And that's why DLI is the integral of PPFD. So you've just summed up the entirety of all the different PPFDs that have occurred over the day. And when you get the area under that curve, that's your DLI. So I'm going to talk to you about the objectives of the 3 studies that I'm doing that are all relating to photoperiods and PPFD, DLI, and biomass accumulation. In my first study, I quantified the effect of different photoperiods, all providing the same DLI on growth and photosynthesis of 2 leafy green crops, which were lettuce and mizuna. And after that, I did another study where I was looking at the development of relative biomass allocation in specific sink organs. In this case, I looked at the leaf blade, petiole, and root, and how these sinks, how the biomass allocation to these sinks changes among 3 related subspecies of Brassica rapa, which is turnip, mizuna, and pak choi. And they're important because they're morphologically distinct from one another despite being genetically related. And I did this study as a comparative study to get some preliminary or baseline data for my 3rd and final study, where I did a similar photoperiod setup where I looked at varying photoperiods with the same DLI, and this time looking at how that affects the relative biomass allocation to those individual sinks in turnip, mizuna, and pak choi. So for study 1, it kind of gives away a little bit. It says longer periods with the same daily light integral increase growth of lettuce and mizuna under sole source LED lighting. So photosystem 2 is what's considered to be the rate limiting step in the process of photosynthesis, because it's sort of first in line on the chain that those electrons have to go through to do useful things for the plants. And the photosystem 2 complex is sensitive to environmental stimuli, especially things such as temperature and light. And a related term is the quantum yield of photosystem 2. And this is a measurement of how efficiently photosystem 2 is able to capture and use those photons for photosynthesis. And it's since photosystem 2 is the rate limiting step, it's a good overall measure of photosynthetic efficiency in plants. And this is just an example, a reminder for everyone from their textbooks about the process. PS2 is all the way on the left, and it's kind of first in line as far as the process for a photon being intercepted. And PS2 is responsible for then shuttling electrons through the electron transport chain. Under highlight conditions, the reaction centers in PS2 will all be occupied and with electrons, and they're not able to as efficiently capture new electrons that might want to jump in there. So this leads to an inefficiency. So if photons are continuing to be absorbed, but PS2 is being overloaded and it can't shuttle all of those electrons into the electron transport chain fast enough, it has to open the release valve, and instead it ends up shuttling some of that excess energy from those absorbed photons, either dissipating it as heat or re-emitting it as fluorescence. And related to that, because of this tendency for PS2 to get overloaded, if you increase the light intensity, the efficiency of PS2 tends to decrease, and that's what this graphic is showing. So what you're looking at is, which is written improperly on the slide. It's supposed to be the quantum yield of PS2 decreases as PPFD increases. And this is a nonlinear relationship. So you have a steeper drop in efficiency when you're at those lower light levels, and it kind of tends to even out as you get into the higher levels. What this means practically is that a lot of advantages can be made if you provide supplemental lighting at those lower light intensities. You can potentially gain a lot more. So recommendations for horticultural lighting, often they can focus on optimal DLI recommendations without necessarily talking about how that light is actually delivered to the plants across time. And this is an example of a Purdue extension document that does just that. In this document, I think the word photoperiod was mentioned once or twice, but it's basically giving a breakdown of just the total DLI that's being recommended for different types of crops. And the theoretical basis for photosynthesis being more efficient at lower light levels has actually been around for quite a while, at least in the 70s and 80s. And studies that have been done where they extended the daily light integral by stretching it out over a longer photoperiod at a lower light intensity. The general result of that is you'll end up with plants that have greater net assimilation and more growth, whether that's often it ends up being biomass as well as larger leaves, greater canopy area, that sort of thing. And some of the examples of plants that have been studied with this particular aspect are radish, sugar beet, beetroot, spinach beet, lettuce, cabbage, canola, celery, tomato, etc. There's quite a lot, but a lot of this hasn't been done very recently. So one of the limitations for a lot of these studies when I was looking through them is because of the technological limitations at the time. One of the ways that they were extending the photoperiod without affecting the DLI is that they would add a low intensity light either at the beginning or the end of the photoperiod. And it was low intensity enough to where the amount of light that it was adding to the overall DLI was pretty negligible. However, it does introduce the possibility that the plant response to these treatments could have been a shade response because of that low intensity light. And it also introduces its non-uniform light intensity across time during your treatments. So that's not necessarily the best thing, depending on what you want to look at. One of the other methods that some other scientists have used to counter this is to maintain a constant light level, and they used different combinations of different lamps. And again, this is partly because of the limitations at the time that these particular studies were being done. They didn't have precision control. They didn't have dimmable LEDs. So they had to use different lamps that happen to have different intensities and combine them to try to get their desired range of PPFD that they wanted. So it's a bit cumbersome. You know, the technique is a bit cumbersome. And again, because they're using different lamp types, this introduces non-uniform light spectra into your treatments. And I even read at least one study that actually combined both of these methods, where they were using different lamps and they were also using low intensity light at the end to extend the photoperiod. So it's just a lot of variability. So one of the pressing need here is that we need more current research using our state-of-the-art technologies that we have access to now that are going to be more relevant to what growers are actually doing today. And like I mentioned before, we now have dimmable LED lighting fixtures with programmable control systems, and we have better precision and control over the timing and intensity, spectral options, and also with the improving efficiency of LEDs, it's becoming more economical for growers to actually use this lighting, which may not have been the case 20 or 30 years ago. So in this study, my objective is to quantify the effect of different photoperiods while all providing the same DLI on growth and photosynthesis of these two crops, like I mentioned, lettuce and Mizuna. And special reminder that I am keeping the DLI the same for all of these photoperiods. Applications for this is that if you can provide light at a lower intensity, then you need fewer light fixtures to provide that light at any given point in time. So that saves growers an upfront cost right away, because they don't have to purchase as many lights. If this produces faster growing plants, then they can also potentially shorten crop cycles and increase their profit, because they're producing more biomass per dollar spent on their electricity. So this was conducted in a growth chamber. I started my crops from seed. This is just a quick overview of the environmental conditions. I'm not going to spend too much time on it, because it's got a lot to cover. But the Mizuna was harvested after 30 days and the lettuce was harvested after 41 days. And this was based mostly on visual appearance of how big were the plants getting. And because we weren't necessarily going to do a direct comparison for the end harvests between them. And we tested 6 different photoperiods ranging between 10 and 20 hours. And we adjusted the light intensity in each treatment using PWM control, which is pulse width modulation. This is an overview of how that relationship between photoperiod, PPFD, and DLI looks. So different photoperiods, different PPFDs, but when you do the math, it all adds up to the same DLI. This is an example of one of the racks that we used. We had 3 racks in a walk-in growth chamber, each rack representing 1 block for the experiment. 15 plants were in a tray and per treatment, and each rack could hold 6 different sections representing period treatment. And the lights could be individually programmed in each section. And we used physical barriers to block light leaks. And this was a randomized complete block design. Like I mentioned, there are 3 blocks, and it was replicated twice in time, once right after the other. So before our final harvest, we collected light interception percentage data, which you can see in the image there. That's an example of the light sensor bar going underneath the canopy. And that's how the light interception percentage is calculated. We also measured chlorophyll content index on the leaves and the quantum yield of PS2, which I just mentioned before, using a chlorophyll fluorometer. And we also took photosynthesis light response curves using the CIRUS-3 leaf photosynthesis system. And that looks at how is the plant responding to a series of increasing light levels. And our post-harvest data that we collected was leaf area and above ground biomass. We analyzed all of the data with mixed models, looking at effects for species, photoperiod, block as a random effect, and any interaction effects if they were there. And the light response curves, once they were generated, I fitted the curve in signal plot using a exponential rise to a maximum. And from that, we were able to from those equations, we were able to calculate the net photosynthesis, maximum photosynthetic capacity, and the maximum quantum yield of CO2 fixation. And so this is the first set of graphs. We're looking at the light interception data for lettuce and Mizuna. Lettuce is on top, Mizuna is on the bottom. So we're looking at, as we are increasing the photoperiod, so lower PPFD but the same DLI, as photoperiod increases, both species had higher percentage of light intercepted. So that's meaning that the leaves that they are putting out are capturing more light than in the longer photoperiod treatments than the shorter photoperiod treatments, which is suggesting either there's a morphological thing going on or an orientation thing with the plants that they're able to capture more light. And there was no change in leaf area, however. So if they were intercepting a higher, more percentage of the light available to them, but they didn't change in leaf area, that suggests that maybe the leaves have changed shape without changing area. And there are some other studies that have shown that in similar conditions where you expose some plants to low light conditions, one of their shade adaptive responses is that they change the shape of their leaves. So some plants will get longer leaves in the shade, even if the overall area doesn't change. This is an example of some notable result, the visual result that we noticed with the lettuce plants. On the far left, you see plants that were grown in the 10-hour photoperiod, so that's the highest light intensity, compared to plants grown in the 20-hour photoperiod with the lowest light intensity. On the top, you have plants taken, photos taken 21 days after planting, and on the bottom, you have plants, photos taken 41 days after planting. So because the difference was so striking, I did collect some data on the leaf angle of the 10-hour photoperiod compared to the 20-hour photoperiod lettuce plants. Surprisingly, though, there was no significant change in leaf angle, at least not at the time that I was able to collect the data. I think that if I had been able to collect it sooner when the plants were still a little younger, I think those differences may have been more pronounced, but they seemed to go away a bit as the plants got older. You can also notice on the bottom photo that the plant on the right, which is under the longer photoperiod, is more compact looking, and it's even forming a head, while the one on the left, which is under the highlight, is not. They were also visible. The color was also visibly different. The plants grown under the highlight conditions had yellower leaves, which I will talk about. So, and this is also an example of something that we noticed. I didn't take numeric, quantified data about this. I didn't look at the frequency, but I did make note of the fact that tip burn was present in all of my photoperiod treatments in my lettuce. And tip burn is typically attributed to a localized calcium deficiency on the fast-growing areas of leaves, where if they're not getting enough airflow and to allow the leaf to transpire and to move that calcium up into the leaf tissue, then it can get those localized deficiencies, and that's what causes tip burn. So it's possible that we may not have had adequate airflow. We did have airflow in the chamber, but it may not have been angled down, facing directly down into the into the rosette of the plants like it may have needed to. So this is the chlorophyll content index, and as the photoperiod increases, both lettuce and Mizuna have higher chlorophyll content in their leaves. And in this example, this is from the second experiment, we also noticed that the chlorophyll content index increased over time. So we're looking at multiple days that this was measured over time, and the overall trend of higher CCI in the longer photoperiods it persists. Although the getting higher over time was not something that we noticed in the first experiment. This was just the second experiment that had that. So now we're looking at the quantum yield of PS2. So as I mentioned, this is the efficiency of measure of photosynthetic efficiency. So as you extend the photoperiod, and in this case, it's being graphed against rising PPFD, because this is going to be more more of a response to the light intensity than it is to the photoperiod. So longer photoperiods with lower light intensity have higher quantum yield of PS2. In this graph, it's showing the opposite, because as the light intensity is increasing, the quantum yield of PS2 is going down because the plants are making, the photosynthesis is becoming less efficient. So in the case of this graph, if you think of the short photoperiod treatments being on the right-hand side and the long photoperiod treatments being on the left-hand side, that'll make a little more sense. And also lettuce was more efficient than Mizuna. So this is the photosynthetic capacity that we got from the light response curves. And there was no change in Mizuna, but there was a decrease in photosynthetic capacity with lettuce as the light intensity was increasing. So it's possible that lettuce just may be more sensitive to this than Mizuna. Mizuna might be better adapted for higher light conditions, and maybe it didn't affect Mizuna as much because there's no change. Now we're looking at the above ground biomass on the top graph, comparing lettuce and Mizuna. This was pooled results from both of the replicates that we did. So this is the biomass data from both experiment 1 and experiment 2 graphed together. And we did see a positive correlation that as you extend the photoperiod, the above ground biomass for both lettuce and Mizuna increased by about 9% for lettuce and about 11% for Mizuna. And at the bottom is something interesting that I noticed, that when I was processing these Mizuna plants, some of the larger plants had a swollen root that kind of looked like a tuber. And so I went ahead and took the tuber biomass measurements for that as well. And that was also significant. So Mizuna had both above ground and below ground increases in biomass. So in conclusion, longer photoperiods with lower PPFD will give you plants with a higher quantum yield of PS2 and all two species, sorry about that, and increased chlorophyll content index in all species, and increased light interception in all species. Yes, all these are supposed to say 2 species, not 3 species, and increased dry mass. So the conclusion from this is that this is supporting the fact that photoperiod and PPFD and DLI are all interrelated to one another. And you can't really study one without taking at least one of the others into account and then maybe controlling for the third, because they all have simultaneous effects on each other. So the practical applications for these results are for crops that can tolerate those long photoperiods. If you spread out the DLI over these longer time intervals, you can get better growth, and plants may reach a marketable size sooner, and you can lower your cost of lighting by purchasing fewer fixtures. So that's my first experiment. So I can take a few questions if anyone has any immediate questions. If not, I'll move forward. So the second study, like I mentioned before, is it's tying into my third and final study. I wanted to do this study in the greenhouse before I did my third study because I wanted to learn a little bit more about how are these particular plants that I'm interested in, how are they actually differing from one another, and how are they differing over time as they develop. So this is biomass allocation in 3 subspecies of Brassica rapa grown hydroponically in a greenhouse. So a pretty amazing plants, in my opinion. They consist of a lot of agriculturally important crops, as well as some agriculturally important weeds, and a great variety of plants with a whole lot of different parts that you can eat. And among these is a species which is the turnip, which is Brassica rapa. And I got interested in this because Mizuna, which was one of the other plants that I'd worked with before, is actually a subspecies of Brassica rapa, even though it doesn't quite look like a turnip, but it's closely related. So that got me thinking a little bit more into all these subspecies and why do they look so different? So here's an example. This is turnip. This is Brassica rapa subspecies rapa. It has a swollen tuber, which is a combination of both hypocotyl and root tissues, and pretty much non-existent epicotyl. And it's primarily grown for its tubers. This is Mizuna, which I had worked with before. This is subspecies Niposinica. It's from Asia, and it's grown primarily for its leaves. So it's eaten as a leafy green. This is pak choi, which is subspecies chinensis. This is also another Asian subspecies. And this, it's more compact in its growth. It has shorter, more compact leaves, and it has these thickened, swollen petioles, which are very distinctive. So obviously, these plants are all different, very different looking from one another, yet they're closely related. So that indicates that there are differences in resource partitioning that's going on during their growth. And as I was doing a little bit of background reading, I was unable to find any comparative studies that other people had done among these subspecies. So that was the motivation for me doing this research. So one of the major papers that I was able to find was a field study with turnips. It just kind of looked at how do turnips behave and what do they do while they grow. And so the major carbohydrates are glucose, fructose, and starch. In this particular study, sink filling, which is the tubers, initiated around 43 days after planting. And the starch levels in the leaves decreased rapidly as the root glucose and fructose were increasing. So there's an inverse relationship and maybe a competitive relationship between roots versus shoots. And eventually the root biomass did exceed the leaf biomass by around 48 days after planting. So the objective for this study was to do a similar kind of study to that one that I just mentioned, where it involved repeat harvests over time. So I wanted to look at the development of these sink organs, which were leaf blade, petiole, and root, and their relative biomass allocation among these three subspecies. And my main motivation was I wanted to find some baseline data for my next and final study. So I did this in the greenhouse. I started my crops from seed. These are just some quick environmental parameters for the greenhouse. And I used calcined clay called arsenite. And this was under natural light conditions, so no supplemental light for these. So these are just the varieties that I used. The Hakurai turnip, Mizuna is just Mizuna, and the Asian Delight pak choi. And they were planted in 10 centimeter pots filled with the calcined clay media. And this was another randomized complete block design with 15 pots per tray and 2 trays per subspecies per bench. So each bench, I had a total of 5, and each bench is representing 5 blocks. And each of these pairs of trays were randomly positioned within the bench. And then they were sub-irrigated with the fertigation solution. And this is just an example of my plants in the greenhouse. The white paper around the edges of the pots is some coffee filter paper to keep the arsenite in place without falling out of the bottoms of the pots. So again, I took repeated measurements of the chlorophyll content index, took measurements once a week, starting from 15 days and all the way up to 56 days after planting. And for each measurement, I sampled 6 plants per subspecies per block. And then I performed 5 sequential harvests, destructive measurements, once a week, starting from day 26. And 6 plants were sampled for that as well. And the measurements that I took for the harvest were leaf area, total fresh mass, total biomass, and the individual tissue biomass of the leaf, petiole, and root. And these were also analyzed with mixed models analysis, this time with doing repeated measures, looking at effects of the subspecies the tissue type, the time, block as random, and then any interaction effects, and use Tukey's HSD for any mean separations. So here's the results. This is the chlorophyll content index over time of turnip, mizuna, and pak choi in the greenhouse. So all three of them increased and then kind of started to taper off toward the end. And pak choi had the highest chlorophyll content index at all times, and then there was no difference between turnip and mizuna. And this is pretty consistent with the way that pak choi physically looked when we were in the greenhouse. The leaves did appear to be darker than mizuna or turnip, although they also appeared to be thicker as well, which I'll mention a little bit later, can affect the chlorophyll content index readings. This is the total fresh mass on the top. So over time, we did not see any difference among the subspecies. They were pretty similar to one another in terms of total fresh mass. They all increased over time, which all that really tells you is that plants grow. And then they also increased in total biomass over time, but they did separate out when it came to total biomass. So pak choi had the lowest total biomass, and turnip and mizuna were about tied for having the most biomass, except for at one point in time at 48 days after planting, there was a difference between turnip and mizuna, where turnip was lower than mizuna. This is the moisture content that was calculated from the fresh and dry mass. So what I find interesting is they're all kind of doing something a little different here. So turnip increased, mizuna decreased, and pak choi increased but was sort of leveling off. And pak choi also had the highest moisture content, which I find kind of interesting that it had the highest moisture content and the lowest total biomass, which I guess would kind of make sense. If all that water goes away, then what's left is not going to be as much. And this is the comparison of the relative biomass between those three tissues within and among the three subspecies. So turnip is on the left, and you see that among all three species initially, leaves are taking up the largest proportion of biomass in all three of them. It's leaves first, and then it's petioles, and then roots take up the least. And it's pretty similar for all three of them, which I found interesting because if you look at the seedlings when they first sprout, before they've put up their true leaves, the seedlings of all three of these subspecies look pretty much identical. You can't tell them apart. And the seedlings look similar, but over time they start to change in appearance. And this is pretty much matched by these graphs. So what's interesting in turnip is that the roots clearly take over time in the turnip, and eventually comprising a little over 50% of the total biomass in turnip, which is not what you see in either mizuna or pak choi. Both mizuna and pak choi, the mizuna leaves remain the primary biomass for the whole time, even though petioles are slowly increasing. Not as much as the petioles increase in the pak choi. It got up to the point where it was about equal parts petiole and leaf for the pak choi. But in both mizuna and pak choi, their roots remained very minimal, somewhere around 6% for pak choi and 8% for mizuna for the relative biomass. And here's the results for the leaf area on the top. So this is the leaf area at each harvest. And mizuna had the highest leaf area by far, and it had the lowest specific leaf area. So specific leaf area decreasing over time is telling us that the leaves are getting thicker. And as I mentioned before, when leaves get thicker, it can affect the CCI readings. So the way that the light absorption and reflection works for the meter for the CCI, it can give you a reading thinking that there's a lot more chlorophyll in the tissue than there might be in terms of absolute concentration, because it's looking through a thicker leaf tissue. So the specific leaf area is decreasing over time. So what's interesting about this is that as all three of these subspecies are seedlings, they all have very similar thin leaves, meaning that they're spreading out their biomass over a relatively large leafy area, and that this is becoming less important over time. And I also mentioned that Mizuna having the highest leaf area is interesting because it also has the lowest specific leaf area. So it has a lot of leaves, but it's also the leaves aren't spreading out as much as, or they're not as thin. They're not being spread as thin as the other species. And this is a graph looking at total biomass per plant as a function of the total leaf area per plant. And so because Mizuna had the most leaf area and the most biomass, it's coming up kind of far to the right. However, if you look at the angle of the slope of that curve, you'll notice that turnip actually has the highest biomass per unit leaf area of either of the three. And so pak choi has a pretty similar slope to Mizuna, but turnip actually, you could say it outperformed them if it comes to biomass per unit leaf area. So, and that's probably because turnip was very busy filling its roots. So it ended up with a lot of biomass in its roots without a whole lot of leaf area to show for it. So as I mentioned, the subspecies, all three subspecies, they started off pretty similar in a lot of different ways, but they grew more different over time. And the most distinctive of this was that sink filling activity in the turnip roots. And what we can infer from the information about the leaf area, the specific leaf area decreasing over time, and looking at the biomass per unit leaf area, is that it seems like these young plants are investing most of their biomass early on into creating leaves, which suggests that it matters to them to put out a whole lot of leaves as early as they can, because that's going to be how they're going to get their light. And that light is how they are going to grow. And that sets the stage for them later on. So investing early into leaves helps to maximize their leaf area and light capture for photosynthesis, which can increase their growth. And this is something that we have seen in another study, another lab mate of mine that was doing a greenhouse study and also looked at leaf area in seedlings, and found that in treatments that had low light levels, seedlings did benefit from creating larger leaves, larger, thinner leaves. So these differences in this particular study were genetic. I wasn't doing an environmental manipulation in this study. I was just looking at differences among and within the subspecies. So the question then is, what environmental cues might alter sink strength of these tissues? And that dovetails into my last study. So before I move on, does anyone have any immediate questions? If not, I will continue.
Hey, Shane, I just submitted one in the chat. I can read that out loud, though. I was just curious if you think the species would respond differently if they had different fertilizer concentrations supplied. I realize that was out of the scope of your work. But for example, anecdotally, I find bok choy has higher fertility needs than Mizuna when I grow them in hydroponic systems.
That's interesting. I don't know specifically for these plants. I mean, I know in general that, you know, if you have a fertilizer deficiency, it can very well affect growth of plants. Because I did another boron deficiency study once with tomatoes that definitely it reduced their biomass and it reduced their height just on a very small trace amounts of boron. If they're lacking it really affects them. So I wouldn't be surprised if you did find that bok choy might be behaving differently and might have different nutritional needs than these other guys. You know, the thing that I've learned is that they say that these are all part of the same species, but they're actually very different from one another in a lot of ways. So obviously their sink physiology is different. clearly their sink physiology is different. So as much as nutrition would tie into that, I wouldn't be surprised.
Sure, sure. Cool. Thanks for your response.
Thanks.
Shane, can you
Yes.
hear me? This is Leo Lombardini. I have a question about your first experiment. I was wondering, what do you think the main obstacles for producers, greenhouse growers to adopt some of the technology that you described? So basically distributing the light across different photo periods. And also based on your experience, how many of them have already transitioned to LED lighting and how many are still using high pressure sodium? Because from what I gauged, I mean, there's the LED lighting are still more expensive. Obviously, long term, the payoff, the return on investment is higher, but obviously there's always that initial investment that can be steep for some of the producers. What are your thoughts on that?
So I'm definitely not an expert, but it's my understanding from what I've read in time that I've been in Mark's lab is that the efficiency of LEDs, while it is still improving, it's still not at the point where it's always going to be economical for growers to use LED lights for supplemental lighting. I think it's still at the point where it's most economical if you have really high value crops, like herbs and medicinals, fresh leafy greens that take that command a premium. But if you're looking at something, say growing wheat or one of the large staple field crops, it's not going to be economic. So from what I've read, I think the economics side of it, getting that side of it to work for a particular crop is still a big limitation. Growers have to think about, is it actually worth it for me to invest in this technology and to pay for that electricity if the gains that I get from it, are those gains actually going to pay for the investment? And that is actually an area that the LAMP grant that I'm a part of in Mark's lab, that's one of the things that they are looking at, is trying to find better ways of working out the logistics of the models, crop models for production, as well as how economics plays into that, looking at variable electricity rates and all that to give growers tools that they can actually use to determine for themselves and to decide for themselves what's going to be best in my particular circumstances. So I hope that answers your question.
Yeah, thank you. It does.
Hey, Shane, I had a question for you real quick.
Sure.
Sort of a follow-up to Leo's question, where I'm sure someone in your lab has gone through and done the economics of like energy expenditure and usage for the LEDs. So you've had a really good support for an intervention where lower PPFD over a longer photo period is going to create more leafy biomass. So how does that work with the economics and the energy expenditure? Does that perfectly scale down where you've got the LED working at 30% capacity for longer and it's the same energy expenditure it costs? Or is there some sort of like threshold expenditure for having the lights on for longer that you're now spending more electricity? Has anyone like looked into the economy of that?
I'm not entirely sure if I understood your question completely, but I do know in Mark's lab they are doing quite a lot of economic calculations to determine, like you said, it's looking at the costs of the electricity under the very specific circumstances that growers would be using it. And one of the things that they're looking at is variable electricity rates. So that is something that comes into play with light intensity. If you are using light at a low intensity, your rate of usage is lower. So you can hypothetically, depending on how you play it, if you're dealing with variable electricity rates, you can potentially tweak your rate of electricity usage in times when the electricity either costs more or less for you to use it, and you get out ahead economically. And the complication of that is, how are the plants going to react to it? You know, if electricity is really cheap and you want to blast them with a bunch of light because it costs you less to do it, are the plants going to actually like that? You know. So, and I think I see some chats. I'm reading the chat messages. Light efficiency, Mark said yesterday, would be equivalent to every light intensity with the lights we use. Okay. Yeah. So yeah, if you're not taking variable electricity into account, I think the simplest model is that you would be using the same exact amount of electricity to power the lights at, say, 200 micromoles for 20 hours as you would to run them at 400 micromoles for 10 hours. So it's costing you as a grower, it's costing you the same amount of money. But if you do it in the 20-hour photo period, you're actually going to get more biomass out of your plants. So you're gaining from that, assuming the plants don't mind having a 20-hour photo period.
That's perfect. Thank you.
All right, I'm going to move on to my final study. Back on. Okay. Okay. So here we go. So in this third study, I'm looking at biomass allocation of these same three subspecies, but now I'm looking at it in photoperiod treatments. So I want to do a similar photoperiod study where I'm spreading the DLI out over a longer period of time with lower light intensity. And from what we already know from prior study, both within our lab as well as, you know, quite a few past research and years ago, is that generally you get better net assimilation and more growth, with a bunch of different examples here. And so the limitation that I found when I was reading about this is that most of these studies that have looked at varying photoperiods with the same DLI, they focus either on total biomass or above ground biomass only, which works okay if you're working with lettuce, you know, and you only care about above ground biomass as a single entity because you're chopping off the head and that's what you're selling. But I was interested in wondering, can you manipulate relative biomass? And I couldn't find very much literature about that. So that was the basis for this study. So what I was able to find in a species that's related to the plants that I was looking at is radish. So there was a study that I found working with radish where Marcellus noted that the relative biomass of tubers in radish increased with increasing irradiance. So in this case, I think that this is just the overall greater light that the plant was receiving, which is not quite the same thing as the experiment that I wanted to do, but it's close enough, saying that there is a light response that the radish is having, and it is changing its biomass allocation in response to the lighting environment. There's another study from Kraker that did look at the same DLI. So when they grew radish, comparing a 16-hour photo period versus an 8-hour photo period with the same DLI, the 16-hour photo period plants, they initiated their tubers earlier. And so by the end of the study, those plants had accumulated a larger proportion of tuber fresh mass. They didn't look at dry mass in this study, but the fresh mass was different. So they attributed this, they said that the rate of sink filling itself was the same between treatments, between the 16 versus 8-hour photo period. What made it different was that the radishes in that 16-hour photo period got a head start, and that's what made the difference. So again, as I was looking, I couldn't find any comparative studies of either total or relative biomass allocation among these three subspecies. So what I was curious is, if those differences exist in a radish, are there potentially other different, are those differences, do they exist in other related subspecies, other related plants, such as Brassica rapa? Will they display that same alterations in biomass under different photoperiod treatments? So the objective for this study was to determine just that, whether we could vary the photoperiod with the same DLI and have that affect the relative biomass allocation, specifically looking at the leaf blade, petiole, and root tissues in turnip, mizuna, and pak choi. So I hypothesized that since we already know historically from other studies that generally, this is true, longer photoperiods with lower ppf will generally result in higher overall biomass. But if I'm going to assume that turnip is similar to radish, then I would hypothesize that I'm going to see a similar relative change in turnip. So I wanted to see if there would be an increased proportion of root biomass in turnip the same way that there was radish. And I expected there to be little or no change in the relative root biomass in mizuna and pak choi based upon what I saw in the greenhouse study, because roots make up very, very little of their overall biomass. And just a reminder, these treatments all have the same DLI. So the applications for this research is, if we can manipulate sink strength, then we might be able to get plants to invest biomass in high value target tissues that we actually want to sell and harvest. So we could potentially improve the crop harvest index, and which would provide more profit for growers. So this study was conducted very similar to my first photoperiod study in the growth chamber. It was in the same growth chamber. These are the environmental conditions, and I harvested all the plants between 35 and 38 days after planting. And I did do a little bit different as far as the photoperiod treatments go. I wanted to get up to 24 hours. So that's why it's ranging up to 24 hours. We did have a sixth treatment of 10 hours, but there was a technical glitch and that one got messed up. So we're only reporting the data for these 5 photoperiods. So they range from 12.8 to 24 hours. These are our light intensities, and showing that it still all comes out to a DLI of 16. And we chose the 16 moles as our target DLI because that's something that some other research has determined to be the optimal high-end DLI for lettuce growth. Although, like we found, sometimes it can cause tip burn when they've got that much light. So this is, like I said, it's very similar to the prior study, but I had a slightly lower planting density or potting density here. I only did 10 plants per tray instead of 15 to give them a little bit more space so they weren't so crowded. And this was 5 photoperiod treatments, randomly positioned on each rack. And I only ran this as a single replicate, although I had 3 blocks. And pre-harvest, we took early projected canopy area images. And I wanted to explain a little bit what that is. The picture that you see on the right is an example of a photograph taken from our top-down canopy viewing system that we have access to in our lab. And this is actually an image of the fluorescence coming off of those leaves from the chlorophyll in the leaves. So this is not a true color picture. This is actually taken with a bandpass filter. So it's what you see in the photo is actually the light that the plants are emitting. They're glowing. Those are glowing plants. You just can't see it with your naked eye. So, but this is a cool technology though, because it allows you to do image analysis pretty easily. And you can count the pixels, and then you can gather up a pretty accurate measurement of what the canopy area is for these plants. Post-harvest, between 35 and 38 days, we did a final leaf area measurement. That's using the conventional leaf area meter. And we measured the total biomass and the relative biomass of the leaf, petiole, and root. And the canopy images were analyzed using ImageJ software to get the pixel counts. And then once I had the canopy sizes, I then applied the mixed model analysis to those as well as my other data. And we were looking at effects of subspecies as well as tissue type, photoperiod, block as a random effect and any interaction effects. And we use 2p's HSD for mean separations. This is just an example of what the plants look like in the chamber. I think they're very pretty. They look fantastic. So on the left are the turnips, which are quite happy growing in relatively small pots. And pak choi is in the middle, and mizuna is on the far right. So these are pretty close to being adult plants, mature plants. So you can see how different they look from one another. So it's very distinctive. You can't mistake them for each other. Here's the results of the total biomass. So this was kind of surprising to me because I thought that I had conducted this experiment pretty similar to the first photoperiod study in the same chamber, but this time, as photoperiod increased, we did not see a difference in total biomass in any of these subspecies. So I was kind of puzzled by that, since in the first photoperiod study, we did get an increase in biomass, and that's been supported by other research from other people, both inside and outside of our lab. So, but what was similar was that turnip and mizuna had about the same biomass, while pak choi had the least biomass. So if you recall from the greenhouse study, study 2 that I just went over, pak choi also had the least biomass. So the patterns in terms of how they're comparing to one another are still there. So pak choi is still coming in last when it comes to biomass, and then turnip and mizuna are more similar to one another. So I was speculating a little bit about what environmental factors might have caused the results to be different than the first time. I suppose it could be random chance, because that happens too, but I was testing a little bit different photoperiods, a different range of photoperiods and different range of light intensities. And the environmental conditions were a little bit different inside the chamber, as they tend to be. It was a little bit warmer inside the chamber this time around. But beyond that, I'm not entirely sure what else could have affected it environmentally. And there's also the potential for genetic factors. So prior to doing this study, I had only worked with one of these 3 subspecies, which was mizuna. And I had assumed that turnip and pak choi were going to behave similarly, at the very least when it came to total biomass accumulation. But it's possible that that may not be the case. There are definitely species-specific as well as cultivar-specific responses to different light intensities. So this has been noted in radish as well as in potato, that long day versus or early season versus mid-season versus late season cultivars will respond differently to photoperiod. This is the comparison of the individual sink tissues. So for the most part, as photoperiod increased, we did not see much change. The only significant changes we did see in relative biomass were in mizuna and pak choi, where the relative leaf biomass decreased. But that decrease, because of the law of proportions, you would expect that if it was decreasing, then that means that the relative biomass of either the root and or the petiole would have to increase to compensate. But we didn't get significant results for those. So that suggests that whatever changes were there in the relative leaf biomass, those changes were so small that it didn't have a noticeable effect on the overall proportions of those other 2 tissues. And there was no change in the 3 tissues in turnip over those different photoperiods. But again, if you compare this to study 2 again, if you take the photoperiod treatments out of the question and you're just looking at the overall relative proportions or relative rankings of these tissues, this is similar to what we saw in the greenhouse study, where the roots are comprising the majority of biomass in turnip, followed by leaf and then petiole. And then in mizuna and pak choi, again, you see where the leaf tissue is comprising the majority of that biomass, and the roots come in last. So in that sense, they are still behaving the way that they did in the greenhouse study. They're just not reacting to the photoperiod treatments the way that I was expecting them to. So this is a graph on top of the early projected canopy area, which I explained before. as it responds to extending the photoperiod. So we did see a significant increase in early projected canopy area in turnip with extending the photoperiod. But that did not translate into other changes in, say, in leaf area or biomass. It was just seemed to be this one parameter that changed. And the final total leaf area in Mizuna was the greatest, and it followed a curving trail, kind of a parabolic trip there, while the pak choi had the lowest final leaf area, which is those two facts are pretty consistent with the visual appearance of these plants. Mizuna has a profusion of leaves. Even though I did not count the leaves in this experiment, but just from knowing Mizuna, it has tons and tons of leaves. And pak choi has relatively few leaves compared to Mizuna, and also has smaller leaves in general. So these graphs on the top, this is final leaf area as it relates to the early projected canopy area. So I wanted to see, is there a relationship between those early measurements that we took of the canopy area? How might that affect the final leaf area later down the line? So we did find there is a positive correlation between both early canopy area having an effect on both the final leaf area as well as the total biomass for these plants. So basically, the more early canopy area you have, the more likely you are to have more total biomass and more final leaf area. Although there are limits to that, because there's species-specific differences here. So for instance, Mizuna has the highest final leaf area relative to the early projected canopy area. But you can also tell that the turnip had invested all of its biomass into its roots. So these species differences, you could say were because Mizuna was making leaves and leaves and leaves and leaves and wasn't doing a whole lot else with itself but making more leaves, while turnip was putting a whole lot of its biomass and a whole lot of its investment into its roots. So you can kind of distinguish that a little bit in the graphs, who's doing what there. But in general, the relationship stands. So these are my conclusions pretty much for all three studies now. If you recall from study 1, we did show that plants produce more biomass under longer photoperiods with the same DLI. And if you now tie that into study 3, where I mentioned there was a lack of photoperiod effect on both total or relative biomass, or most of the relative biomass, when we grew turnip, Mizuna, and pak choi under these longer photoperiods with the same DLI. And there's a possibility that there may be environmental factors or genetic factors affecting this, but I would actually love to get some other people's feedback about what they think may have happened to have this kind of outcome. And now, if you recall the study 2 results, that's the greenhouse study, what was interesting from that was looking at how the seedlings changed over time. They started off with a higher specific leaf area and a larger proportion of leaf biomass early in their development. And now if you tie that into study 3, this most recent study, when you're looking at that positive correlation between the early projected canopy area and the final biomass of the plants, it suggests that early canopy size really is playing an important role in later biomass production. So I think that's a pretty important takeaway as far as when it comes to future research and things that people might want to look at. You know, where is your intervention going to do the most good? And it seems like when it comes to photosynthesis, at least the stuff that we're working on here, affecting seedlings as early as possible and getting them to put out the biggest leaves they can as soon as they can is where the best reward is going to come. So there was another study by another one of my lab mates that was evaluating some greenhouse lettuce cultivars, looking at phenotyping actually. And he also used the same early projected canopy imaging system that I did. And that was correlated with final above ground biomass in these lettuce cultivars. So they make the case that the technique, this canopy imaging technique, could be used for rapid phenotyping to select fast growing crops for breeding. Basically, if you can identify them when they're seedlings or very young plants and say, wow, this plant has a lot of canopy area, it's going to be a big grower later on. That's the benefit for using this technique. So with that, I'd like to thank my committee members, Dr. Mark Van Ursel, Savitri Nambison, and Dr. Neil Natson, as well as all of the fellow lab mates and all the other horticulture students and grad students and assistants and everybody else that has helped me. I hope I didn't leave anyone out here. This was a great opportunity for me going here, and I really appreciate all of the help and assistance and companionship that everyone has provided. And with that, I would also like to thank my sponsors, grant from the USDA. And with that, I'll take any additional questions.
Any more questions for Shane?
I just have one question. Study 1 and study 3 just had only Mizuna in common. I'm wondering if bok choy and turnip also shows an increase in biomass. Have you seen that in any other study? Because those two were newer in your study 3, right?
Yes.
And so I'm wondering if any other lab mates have done a similar study, or is there a reason to believe that those two species may behave differently under longer photo periods and low light intensity?
I don't know. There's been one other student in our lab that worked with turnips as well, but I can't recall off the top of my head what the treatments were that he was looking at for what he was with. So I don't know off the top of my head of anyone else that's been looking at the lighting interventions or any sort of environmental interventions to increase biomass in either turnip or pak choi. So, especially not pak choi, because that seems, I could barely find anything on pak choi really when I was trying to look stuff up about it. There was more on turnip, and there was a fair bit on radish. That pak choi was pretty sparse.
Hi, Shane.
When are you graduating?
May, the first week of May.
This May.
This May, yes.
Oh,
so
okay.
Very soon.
And what comes next?
Oh, well, I have 2 job offers that are on ice right now because of the coronavirus. I've got one of them is I got an extension internship working at the botanical garden with the Georgia Extension, but not sure if that's going to work out yet.
And those 2 jobs, the first one you mentioned, are in the private sector?
As of right now, yes. I mean, well, the internship is one of the jobs that.
Right. Oh, okay, that's one of them.
And then the other job is federal actually. The other job is the National Park Service. So I have a technician position with the National Park Service, which is also on hold at the moment.
Mm-hmm.
So, yeah.
Well, thank you. All the best and good luck.
All right, thank you.
Any more questions?
Shane, I was wondering, I had messaged you in the chat, but you probably didn't see it. So I was wondering what week of the growing cycle you took the canopy area images. I missed that when you were saying.
Oh, if I recall, I think it was 15 days. 15 days after planting. Let me pop back up. I'm pretty sure that's what it was. The early canopy images. I may not have mentioned it in here. I'm pretty certain it was 15 days. I'm pretty sure when it was taken. And then the final leaf area was at harvest, so it was somewhere between 30 and 38 days.
Yeah, I was just wondering when the early ones were taken.
Hey Shane, I have a question for you.
Sure.
So what what's the meaning of the photosynthetic capacity in the experiment 1 in here? Is that a symptom of the photosynthesis light response curve?
Yes, the photosynthetic capacity was one of the things that we were able to get from the light response curve. I may have forgotten to mention that the other two parameters there were no significant differences. So I just reported the photosynthetic capacity. So in the lettuce, the capacity decreased as the light intensity increased. So that was suggesting to me that the lettuce was not as well adapted to handling the high light situation. But someone else can correct me if I got that wrong.
Thank you.
I mean, I would agree that the lettuce doesn't like the high light.
Shane, this is Neil. I was just trying to brainstorm other differences between experiments 1 and 3. And I guess plant spacing was another difference, right? It was a higher density in the first experiment.
Yes, it was a higher density in the first experiment, and the plants kind of crowded each other. Crowded each other both within the tray and adjacent trays. So that's part of why I decided to go down to 10 pots per tray instead, to kind of give them a little more space to breathe so they weren't interfering with each other.
Sure, and that certainly makes sense. I wonder if there could be some, I can't think of a physiological reason myself, but maybe there could be some reasoning where the plants at higher density, like in your first experiment, showed this response to stretching the light over a longer photo period, but for some reason, when the plants weren't packed together in the third experiment, then they didn't show the response.
Yeah, maybe. I mean, maybe when they're packed together, maybe there was some sort of additional shade response that some of them were experiencing because they were getting shaded out.
Could it? Could it also be a change in transpiration when they're packed together like that? They form their own little microclimate.
No,
more spread out, you're going to get more transpiration, I would assume.
Yeah, I hadn't thought of that. One of the other differences too is that in the second photoperiod study, which is study number 3, I used that arsenite clay as my potting medium because it's easier to wash it out of the roots. Although easy is a relative term. But the first photoperiod study, they were planted in regular potting mix. So I don't know if the potting media could have affected something. I know, actually this goes back to Neil's question about nutrients. I know, Laura, you've said before that when you've grown corn in a greenhouse, adding the arsenite to the potting media for the corn helps to prevent an iron deficiency in the corn that it seems to only get when you grow it in the greenhouse.
It only gets it in peat.
Oh, in peat.
If you add field soil, that also remedies it.
Okay. So if that's the case, then maybe the arsenite itself has some sort of maybe it's leaching something out that could be affecting the growth of the plants.
It would definitely have a different pH, probably. Would.
Okay, is there any last minute questions? We are way over time, so I don't want to keep people too long. Okay, then I just want to say thank you to everybody through this very strange seminar series. Good talks, but yeah, a little bit unusual. Thanks for all the speakers that were willing to do it. There was a couple of speakers whose seminars we had to cancel, John and Emma, and we'll reschedule that probably in summer. So look out for some emails about upcoming seminars. Thank you, everybody. Stay safe.
Thanks for coordinating, Cecilia.
Yes, thank you.
Stay safe, everybody.
All right, see you guys. Good job, Shane.
Thanks.